Hi, my name is Anderson. My colleague Jet was unable to be here due to emergency surgery for a broken leg, but he participated in the rest of this project. So I will be representing Team Plastic Patrol. Did you know that 8 million tons of plastic enter into the world's oceans each year, yet 99% of them are invisible? Even more shocking, there are 24 trillion pieces of microplastics found in our world's oceans. The study of plastic waste is not new, but the question about microplastics smaller than 5 millimeters are only just beginning. Pioneer studies suggest negative impacts on cells, and last month, a study in Amsterdam found microplastics in human blood for the first time, confirming that polymers pass across membranes, and ecotoxicologists fear this could trigger disease in living organisms. Microplastics allow a path of concentrated chemicals through the marine food web, and they absorb carcinogens like DDT and PCB very well. These contaminants are circulated through the ocean currents, and a major concern would be the impact on endangered species like the hawksbill sea turtle that nest on beaches in 60 countries and whose hatching rate is at a global decline. The United States nation's Sustainable Development Goals list the impacts of microplastics on biodiversity as one of the most critical endangering issues on our planet. Methods to remove microplastics from waterways are urgently needed. Our research was extensive and included professional journalism, TED Talks, books, videos, and interviews with experts. We worked with the Institute of Environmental and Human Health and selected algae as a possible accumulator for our microplastics. Based on research, we designed 3 experiments to test whether a living resource could effectively mitigate microplastics in marine and freshwater ecosystems. And we investigated the impact of microplastics on the temperature of sand that could affect nesting sites for sea turtles. Tests were run multiple times and with microplastics present in sand, and a 4-degree decrease in temperature occurred and could result in a negative effects on the sea turtle's viability and the gender determination of the eggs. Next, 20 species of marine and freshwater algae were grown in 20 habitats in the lab. Microplastics then were made and treated with non-toxic fluorescents so that the plastics would glow under a black light for greater visibility. We added the plastics into the algae habitats and hypothesized that algae would bioaccumulate the plastic particles. 4 weeks of observing under microscopes shows 2 species of marine algae, Polysiphonia and Calithamnion, and 3 freshwater species, Vaucharium, Peridinium, and Lingbia, mitigated 35 to 60% of the microplastic pollution. This was a great victory. The novel use of algae for mitigation has been shared through our website and educational displays in coastal museums across our state. We reached out to Bonnet Springs in Florida and will provide data for their displays as well. We partnered with Texas Tech and designed a cell viability test for hawksbill sea turtles for 2023. This benefit will be life-changing. Algae can be a great weapon used to mitigate microplastics and is far more effective than other methods requiring more manpower and economic resources. We are glad to work beside pioneers in answering these tough questions, and as God's stewards of our environment, we will do our part. Our project's potential is to create a sustainable solution to one of the most unique and pressing issues on our planet.